Okay, so welcome back to session 2 of this NIAB Soft Fruit Day. Hopefully you found the first session useful. Just to run through the presentations that we've got coming up for the rest of this morning. Papp-Rupar is back with us again. We're working him hard. He's going to talk a little bit about work that we're doing at East Smalling on the efficacy of biocontrol agents for disease control in strawberry. We then have a couple of talks on breeding and variety development, one from Feli Fernandez on breeding raspberries and blackberries, and then Alin Borleanu will talk about our experiences with morning fruits. Samantha Lynn will join us to tell us about the latest work she's been doing on powdery mildew gene resistance, and Tom Passey will finish the session talking about Mucor and Rhizopus. So welcome, Matt. I'll just stop sharing my screen. And a huge thank you to Matt, I should say, at this point, because he had a very, very busy day last night and was busy putting his presentations together well into the small hours. But welcome back, Matt. I should just explain, we have just shuffled the order around slightly from the original plan because Matt's got another engagement a little bit later, so I've moved him to the top of this session. Matt, good morning again. The floor is yours. Good morning.
Okay, very good. Thank you. So, yeah, we're shifting from recycled coir to crown rot controlling coir, which are kind of intimately interlinked, actually. I'm going to cover again quite a lot of things that we've done at NIAB. Some been done by Shengming already before I started to kind of look into this, and then as more of new things that we've been developing this year and last year. So the challenge, you probably know this challenge quite well, at least if you're a strawberry grower or agronomist, is a crown rot, which can lead up to 20% plant mortality in plantings, even worse in soil than it is in coir. But it's a huge problem in coir as well. And that really declines your yield, and sometimes the quality of the remaining yield is reduced as well. You get it in through infected plant material, which is latently infected. So it's almost impossible to know without destructively sampling, without killing the plant. It's always impossible to know whether it has Cactorum or not. But can also come from untreated irrigation water from ponds that you might collect. You might get some infections in there. And then it very quickly spreads. It grows mycelium around the roots, and then forms this sporangia, which are little cells that produce zoospores, which I'm only mentioning because they swim. They're called zoospores because they swim, and they can swim from one end of the bag to the other, or if you have multiple things connected and they find a film of water, they can swim and spread. So that's why it's so difficult to manage. It's not they spread really well, and they can infect fruit as well, called the leather rot on the fruit if you have a high disease pressure. Infection rate can be quite high in your original planting material, and it varies immensely from consignment to consignment. So this is something that Shengming and Tom Passey did a couple of years back for an AHDB, I believe one of the AHDB trials. And different cultivars provided by different nurseries may have a very different overall percentage of Cactorum. So here what we did, we just took, I believe, 100 or 200 plants at random before they were planted at different farms, and then destructively sampled. So the infection rate varies massively. Some very good, some very bad of the same cultivar. So be mindful of that. In coir system, we had chemical control. Phenomenal, was quite good, was fairly effective, depending on how badly effective your infected or your planting material was, but it's not authorized anymore. Parrot is expected to go. I think we still have it this year, maybe next year, I'm not entirely sure. And then the really important control that you could do is a culture control. So obviously clean plants for the ones who are producing your own plants. There are some growers that they do that. Producing clean plants and clean environment, using clean water and clean substrate is going to keep the crown rot away. You use resistor tolerant varieties, which maybe Alan's going to touch on a little bit later. And then really when you have the infection in, which inadvertently to a small degree you always get, avoid over irrigation to prevent spread. So these Phytophthora need water to spread, and avoid heat or drought stress, because they sort of accelerate symptom expression. So if you forget to put the tunnel sides up and it gets really, really hot for a couple of days or a day, you might accelerate crown rot symptom expression. But one thing is missing here, and that's the biocontrol organism. So I'm going to talk quickly about this. So you're not alone with problems with Phytophthora. Okay, this is a worldwide and a really important pathogen, Phytophthora. There's more than 100 species of Phytophthora that we know, and they most, if not all, are capable of attacking a wide range of plants. And here are just the two: potato late blight and southern ogda. One historically really important and still in pepper, tomato, potato, and other solanaceous crops. And then southern ogda is like an invasive Phytophthora killing oak and to some degree larch as well in the UK and the rest of the world. And people have been working on biocontrol for a very long time for Phytophthora. Obviously, they survive in soil really well. They have this super long-lasting resting structures. They're very difficult to inactivate. So having microbes to compete with them would be one way. And people have found bacilluses, Pseudomonas, Streptomyces, and other bacteria, filamentous fungi, trichoderma, and also yeasts. They found a lot of different mechanisms of how they antagonize or control Phytophthora. So they can control mycelium growth to prevent sporulation or prevent zoospore germination, and some actually trigger systemic resistance in plants, and then plants fight Phytophthora a little bit better upon inoculation of biocontrol. So what happened then a couple of years back is that two sort of, based on this literature, two kind of, or a couple of commercially available products, but not registered in strawberry, were tested. And with yellow, we have T34 trichoderma, and in green, you have pre-stop. And you can see, compared to phenomenal, proportion of wilting plants it can be similar to phenomenal. Proportion of dead plants can be similar, slightly higher, and average yields when using these biocontrols can be comparable to phenomenal. So yes, to a degree, you can decrease crown rot impact and safeguard yields with the commercial products already. But here we applied it, I should say, as a drench. So each plant got a high dose of spores, and it might not be something that you can actually do in the field. So we tried an in-irrigation application, and again, we could reduce plant death, but with both, and to much lesser degree with trichoderma, and to better degree with pre-stop. So we can control with current commercial products to some degree, but it's not as effective or consistent as a chemical controls, and we still need approvals for use. So one of the things that we think it's also need to be optimized is delivery. How you deliver in your crop these biocontrols that are economically feasible and effective we still need to investigate. So this is very preliminary. So, but can we do better than this? Can we do better than this filamentous fungi biocontrol that are available on the market? So we would need to do better. We would need better colonization of our biocontrol. So when you put them on to colonize more of the media, better survival for longer, and then that will produce more consistent and more effective biocontrol. But we can actually start adding other functions on top of that. We can start adding growth promotion. We can start adding protection against the range of pathogens that you see. We can add abiotic stress tolerance, or possibly increasing resource use efficiency by adding the right microbes into the coir. So there is a potential to go from biocontrol to sort of a microbiome approach, where you get many different functions from your plants, from your new microbes put in. So how do we find this? So we think bacteria will be better because they're cheaper to produce than fungi, and cheaper to find and easier to investigate, and they might be better to survive in really high moisture, high nutrient content environment. And they are known to have quite a lot of other plant growth promotion traits that we might want to incorporate. So how do we find them? Right, so basically we start at the root of the problem, which is a strawberry root, of course. And we have, or I should say, I have sampled a couple of years back I have sampled commercial strawberry cultivars grown on site in commercial conditions after the last harvest. That means that the bacteria that I find there were able to survive probably through the season, or were there at the end of the season. And I've collected it from several different experiments just to have a bit more diversity. And you have to focus on something. You can't deal with tens of thousands of bacteria that you might find. So I focused on Streptomyces, Bacillus, and Pseudomonas, which all have known Phytophthora biocontrol and other properties that I want to incorporate in this sort of microbiome approach to control Phytophthora. So we've set the collection up, which means that I've used coir, roots, and root washes and roots to get the bacteria off. And I got about 300 pure strains after a couple of months, which is great. But then what do you do with all these strains? You still have to know which ones to use. So here, Jen and our pathology team really comes in. They use this dual co-culture assays when they put different strains of bacteria against different Phytophthora cactorum crown rot strains, and they tested all 300 versus 3, so there was 900 different assays on media that is optimal for crown rot. So you get crown rot the advantage, and if you can control it when the crown rot has the advantage, you expect to have a slightly better result in the field. And here we just looked whether it stops crown rot growing or not, just plus minus, which weeded out pretty much 90% of bacteria that are isolated. So we now have 30 isolates with potential for in vitro antagonism. So for potentially being effective on the plate for crown rot. And I say potentially because of what you're going to see in the next slide, which is you then take those 30 strains and do some proper assay when you measure growth of the pathogen, growth of the bacteria, and calculate to what degree different bacteria reduce growth on the plate. And this was done in collaboration with a project student from University of Kent, which was supervised by me and Jen. And she did, again, 3 Cactorum strains against 30 bacteria, and did everything in 6 replicates. So now we have we can do statistics on, and we found that the best one are reducing Phytophthora growth by 40%. So not completely stopping it, but reducing quite a lot. And you can see here on the picture, the green one is the 1R37, which will be somewhere on this list. There you go, here at the second best. And these ones are in red because they look like they're stopping it, but when you do enough reps, they actually are not consistent enough, so they're not significantly reducing. So that's great. And now we have top 6 of our bacterial strains antagonize all 3 different Phytophthora that we've tested against, which is good. We have something that looks to work against many Phytophthora, but there's many strains that only work against one, but not the other 2. So there is some strain specificity going on. But then how about other fruit rot pathogens? I said we need to think about where the other pathogens might come in. So we had different pathogens that we tested all this 30 against. This here you just see a top of the table, and again just look at the color. The green is good if the white is no good. So we tested again Phytophthora fragaria, which can happen more in soil than in coir, but can happen. And then we have new pathogens that a PhD student of ours isolated from raspberry, which seems to be up, which is Phytophthora citrophora, cryptogaea, and then phytopithiums. And then macrophomina we see every now and then. That's a problem in soil around the world, but we do see it coming with planting material every now and then. And these are in the same order as before. So you see the top 6 that were before top 6 are still in the top 6. And we have strains that are able, like this R80, R23, R37, which has lovely little names. They're able to inactivate quite a lot of our pathogens, if not all of them. And then we go one step extra is like, how about plant growth promotion? So the same student looked at the auxin production. So the bacteria can produce auxin to induce root growth, which may be a trait that can counter root killing by Phytophthora, or can just increase nutrient use efficiency if you have a better roots. And then phosphate solubilization, she looked as well. Phosphate can be in some circumstances can become unavailable to the plant, and some bacteria can solubilize it into soluble form again. And here in the top 6 again, we have a few that solubilize phosphate quite well, and some that produce small amounts of auxin as well. So there is a potential to use the same bacteria to potentially treat or control many different microbes and many different pathogens, and add some positive growth promotion traits in with the same mix as well. So in vitro, it looks like none of the bacteria by itself will be able to control crown rot to significant degree. So we're going to probably have to mix them. Multiple pathogens can be controlled by the same strains, which is great, and appropriate mixes may be able to provide protection and plant growth promotion. But the real crux is, does it work in your commercial setup? So we set up a field trial where we selected top 4 strains that we tested before. We kind of made sure they don't kill each other straight away, so they kind of at least can coexist on the plates. That are high diversity, so the bacilluses and pseudomonads, and that have the best biocontrol potential against crown rot that we're going to test. And we've inoculated June bearer in virgin and recycled coir to see if different media have an effect on that. And then we compared it to the previous commercial biocontrol treatments, and we mainly assessed yield and plant health. Now, with cactorum or crown rot, unfortunately, what our main challenges are that we get a lot of cactorum when we don't want it, and very few when we put it somewhere. So this has not been inoculated, this treatment here, and these all the others have been inoculated. And you can see that inoculation did not really significantly reduce yield. We don't know why. We inoculated twice every 2 weeks with high zoospore concentrations, and we couldn't really get it working this year. In some years, it works really well. It didn't work this year, so the results are not very conclusive about biocontrol. But at least when we can say that our bacteria here did not significantly reduce yields, because that might happen. You never know. So at least we didn't significantly reduce yields versus the untreated. And there was some small variability between other treatments, but not massive. If you look at the class 1 yield rather than total yield, the situation is pretty much the same. The differences is slightly higher, and now bacteria are slightly better than untreated, but not significantly. If you look at the number of fruit, is the same picture. Bacteria slightly better than untreated, the same as non-inoculated, but not significantly. Berry size. It's interestingly in recycled media we have much higher berry size than in virgin. Why? It's not significantly different, but it's trending towards it. So we don't know why. But again, our bacteria were not worse or better than the untreated, at least not significantly. And then when you look at percent of plants that had symptoms, then it's a strange picture. Which with the untreated virgin has the lowest, as you would expect, but then there's a huge variability in percent of symptomatic plants that we see. And our bacterial strain is not performing really well in the percent of symptomatic plants. But I have to stress here that percent symptomatic plants is, with the background of our low inoculation success, is most likely due to having a few more plants infected from the nursery in that particular treatment by random. The differences are very small and overall not significant. And then if you look at the severity, so are these symptoms very severe, so plants dying, or they're just a little bit wilted or just a little bit stunted, then you don't see any differences in severity really, nothing significant. So this is a bit of a downer. We're going to have to repeat and try to get inoculation higher or get more infected plants. But at least we didn't have negative effects with our bacteria. And we are still looking into a survival of biocontrol as a separate trait to see. And we will need further optimization of our bacteria to get it onto the market, which is product formulation still need to be done, delivery needs to be done, and then we have to see how the colonization and survival. And we don't really exactly know what our biocontrol mechanisms are. So there might be a better mix of our bacteria with multiple mechanisms. And then the big one is going to be towards registration and production of this to get it to your fields in the future, which might be down to people in Bayer or Syngenta to get in to contact them and try to get this over the line. So keep take-home messages like keep or improve your cultural control is always the top, because it's the cheapest is the easiest to do without any regulation, without any chemicals. And then to a degree, commercial biocontrol agents can be effective, but we need to optimize the delivery and get them registered in strawberry. And then this novel multi-purpose biocontrol agents or microbial consortia, we're developing them, other people are working on them, but this bridging the gap between lab and field is still a challenge. So we're still working on that. With that, I'd like to acknowledge especially Jennifer and Jade, who did most of the work that I presented, and all the funders from Worshipful Company of Fruiters who supported this very generously, BBSSC and Overland and other noted here. So with that, I'll thank you for listening and get some questions in the chat, probably.
Thank you, Matt. Yes, just one question here. Did you find any Streptomyces species when you were screening for bacteria, which are known antagonists of oomycete pathogens and commercial products available, but albeit not currently authorized for use on strawberries? So you did you?
Yeah, I found, but what I found, and they were so difficult to work with in my hands. So the ones that I found were growing so slowly, and they were not sporulating. Or at least I'm not an expert with Streptomyces. So for the work that the student did with Bacillus and Pseudomonas, I removed it because I think she would be, she would say she might left our lab if we get them like another 100 Streptomyces that are very difficult to work with on top of her 60 or something other bacteria that she worked. So I have them. We have been looking into that more in sort of try to get a PhD around it, but then it didn't come through. So that's on the back burner at the moment. But yes, definitely, we have found them, and they're there, and they do they are, at least on the plates, have efficacy against crown rot. So there are some, not that many as the other ones though, but there are some.
Okay, Matt, thank you so much for your time. If there are any more questions for Matt, perhaps you could contact me directly, because Matt has got to leave us now. Thank you, Matt, for all your input this morning. Much appreciated.
Yeah, thank you.
We're going to have a break from soft fruit disease for a bit, and we've got 2 talks on variety and variety development, starting with Feli Fernandez, who's known to most of you. We have not had Feli on this platform for a couple of years, so I felt it was time to give her an opportunity to bring us up to date with all the work she's doing on raspberry and blackberry breeding. So, Feli, if you'd like to try and share your presentation with us. If you unmute yourself, Feli, then we can hear you. Just bear with us as Feli's having a bit of trouble unmuting.
Sorry.
Here we are. We've got
once I start sharing my slides, I can no longer see.
I know, yeah, I have that.
And so it becomes a little bit.
Don't worry.
Can you see my slides?
They're just coming up now. Yeah, and if you want to hit slideshow, we're up and running.
Is that working?
All good to go. Thank you, Feli.
Excellent. So I don't need to introduce, obviously, the program very much, because a lot of you would be familiar with it. But just to say that I've been the raspberry breeder at East Moreland since 2008. And we've been doing blackberries a little bit more recently, a lot more recently, since 2017. So I'm going to just give you a bit of a quick rundown of what the talk is going to comprise. I'm going to give you a very quick overview of the history of the program, and that's because when I saw on the title Scott has given me for today's market, it got me thinking of the of the progression and how the program has changed and how the priorities have changed through time. And so then we go on to the current organization of the program and how our priorities have shifted, even within the life of the existing project, and how that has changed with where the funding is coming from, and where the lead is in the varieties. And then I'll say a little bit about our 2 current release varieties and some of the selections that are coming up. So we've now been doing raspberry breeding at Ismoilin for over 100 years. We started with working on European raspberry only, so pure Rubus idaeus. The 1950s was all about introducing the good traits from other things, so desirable aphid resistance, cane disease resistance, fruit firmness, brightness, crop potential. And then in the 70s, we started to see the first results of that hybridization process. So we had things like Molin Leo and the Light were some of the first cultivars that had traits from other Rubus species. And then we came to the 80s to things like Autumn Bliss. There are a complete breakthrough for Papp-Rupar breeding, but also are a mix of 10 different Rubus species in the background. Up to 2003, the program was funded by DEFRA. Then we had an industry consortium that funded a very small part of the program, but sat on an advisory capacity for DEFRA's funding until 2008. Then we had a number of years of transition, was we repositioned germplasm and the selection process to be more industry facing. And we worked with a number of partners during that time. And we arrived in 2015 to what is now the Ismoilin Rubus Breeding Consortium, which runs until 31st of March 2025. These are the current partners at the moment. Lubera, who we work with for the amateur market, Onuba Fruit in Spain, Perfection Fair in Australia, Toby in Switzerland, and then the Greenery in the Netherlands, and in the UK, Chambers, Berry Gardens, and Blaze Plants. That changes from next year, as both Berry Gardens and the Greenery are leaving the program at the end of this year, so a year before the program actually ends. And that's to do with changes in the industry, which is, I suppose, part of the topic of the presentation, and with some of the pressure that growers, certainly from the growers' point of view, that growers had to reduce costs, and therefore how R&D budgets are suffering for some of them. And breeding is the longest term R&D project that you can have, so it's a challenging one. So the way the program is divided at the moment is 65-70% of the program is still raspberries, and 30-35% of the total breeding effort is dedicated to blackberries. And the very first blackberry crosses were done in 2018, so this is a relatively new program, and we only have the first proper trial is happening in 2024. So there will be probably some interesting news next year, but at the moment it's all very early stages, and it's been mostly about selecting new material and getting the characterization of that and breathing it again into the program from what is commercial available out there. The split in blackberries is 50-50 between floricane and pramocane. Floricane obviously having a lot more commercial relevance, but pramocane where we have a lot more work to do to get to where we need to be. In raspberry, at the moment we have about a quarter of the program to be strictly floricane, a quarter to be strictly pramocane, but which I mean early fruiting pramocane that comes into crop within by the end of August, September, and then 50% is double cropping. So these cultivars are genetically pramocane, but they leave sufficient cane for the second year that with manipulation you can play between the pramocane and the floricane season. They're obviously also more affected by seasonal changes. So in any given year, you might not always get the long cane length that you want, and that needs to be communicated with the growers, and we need to work on controlling and manipulating those seasons. The raspberry program involves mostly red raspberries, obviously, but we also have an interest in yellow, orange types, where we're doing some genetic studies as well, and in purple, black types, which some members of the industry have an interest for the derived products out of them. In terms of breeding objectives, obviously we want the raspberries to be perfect. There is no discussion in there, but what constitutes perfect, especially on plant habit, has changed quite a lot. So we used to select Christmas tree looking things, as many laterals, as many fruit in its lateral as possible. But actually those very complicated architectures are now quite problematic in terms of management of the canes, of picking costs, pruning. So whilst fruit quality and shelf life obviously have always been a priority, yield and fruit size, again, are always a priority. Cane architecture and the ease of picking are definitely coming much more to the fore, not just because of the reduction of cost in the staff time at the moment, but also with a view that we might end up having to have mechanized picking. And what makes a person pick more easily would also make a robot pick more easily. And then pest and disease resistance, that when we go through how those priorities have changed over time, you'll see that it's quite a key one. But you will also see it's part of the screening, but it's not as important to us as it once was. Not because we don't want it to be, but because you can only have so very many top priorities. And so we still do quite a lot of passive selection by deselecting anything that shows a strength susceptibility, but we do less pre-screening of the material. And then environmental adaptation, we combine that in the program by having seedling selections. The seedlings are selected on their own roots in the ground. So they grow as the old-fashioned raspberries used to. But when it comes to trials and evaluation of selection, we do it in containers and in a control environment, very much like your own commercial tunnels will be run. In blackberries, fruit quality and shelf life, yield and fruit size are key. Other things like, and in shelf life include resistance to reversion and all other things, but it's also thornlessness is a key objective, particularly so for the pramocane material, where there is not very much germplasm that is both pramocane and thornless. So we are putting quite a lot of effort in increasing that. Now, every time there is a change of objectives or a change of emphasis in the breeding program, I come and talk to the funders and I talk to the industry about turning around an oil tanker. And so I'm showing you the breeding cycle so that you have a bit of sympathy for that, because not because I'm going to take you through the whole detail, but so that you can see the very minimum it would take for growers to see some of the material that we produce would be 7 years for them to complete those very first trials. In a pramocane only material, if you're also looking at floricane fruiting, then you can get up to 9 years before you have an actual feedback from the industry. And so releasing a new variety and changing the priorities in the breeding program takes quite a bit of time, which is why I'm always loath to completely remove any type of material from the breeding program. So for a long time, I was told that I shouldn't bother breeding floricane material because there were other programs doing that. And that would have made double cropping material and some of the exclusively floricane material that we are now in advanced trials impossible to get to. So I'm always loath to completely change direction, because 5 years down the line, there might be a slight change on the other direction. So in terms of how those priorities have been reordered and how it's been driven, I think it's fair to say that up to the that you have like 3 clear periods. Up to the 1980s, the 90s and noughties, and very distinctly from 2010 onwards. So the breeding program used to be led by the breeders only and be curiosity led. So interesting traits, interesting what they thought would be good for the industry. We then moved to a very government policy led program where pest and disease resistance and things like water use efficiency became very important, which is not that they are not important, but that was the key focus. So I think fruit quality suffered a little bit on that time. And more recently, we've been completely industry-led. So in swifting those priorities, it's taken us a little bit to catch up. And now we are looking at material of much improved fruit quality. The research used to be completely built into the breeding program, and progressively that has become a mix of being in the program and project-led. And now it's mostly project-based. So some of the growing kind of Medway projects that we've got at the moment will inform the breeding programs going forward. Some of the work we do for BBSRC or hopefully for DEFRA in the future will also inform the breeder, but they are not within the program. The term of the projects has also varied, and equally, as we were saying before, what are the key objectives? You can see how the ordering of these things has changed, and fruit quality certainly is the absolute top now, which hasn't been until the last 10-15 years. The first stages had a game-changing steps. So you have a new variety that will have a massive change to what was there, whereas now we are very much into an incremental progress and also in finding a point of difference. Sometimes some of the breeding that we do, we're looking for something that is different, not just something that is significantly better, because of how the commercial deals work that you're all very well aware. And that has had an effect on the staffing and how the farm is integrated, and all of that is not particularly important. But I want you to look at the very last row on those tables, which is the number of seedlings that we look at. So we don't select necessarily many more than we used to from much smaller populations. So our selection rate has become a lot more strict. And that's all to do with resource of the program. If the program has the resource to process a lot of material, we can be more strict and we can have better results. Now, I just want to very quickly talk a little bit about some of the material that we've got there in the industry, in case you're not familiar with it. So Molin, these are all this information is available through the Molin Fruits website in the NIAB site, and the little QR codes would take you there as well. Molin Charm is it's been released since 2017, just as Molin Bella. It has had a more difficult time getting established in the industry. It's a very early promocane season. It's not particularly well suited for double cropping. And so it has struggled to fit in the industry a little bit more. It requires more careful irrigation because excess irrigation would make the fruit softer. But it's very pale, it's very sweet, and it's got a very attractive presentation after shelf life. It's particularly good from root blocks, so from mown down, you can keep the pots for a number of years, and it works quite well in those conditions. Molin Bella, which quite a lot of you would be familiar with, and it's becoming more significant in the UK industry. It's growing a lot at the moment in southern Spain. What you find in the supermarkets of it at the moment would be all coming from Anuba fruits in Spain. It's well adapted to double cropping, so it does, excuse me, it does reasonably well as a long cane. And in fact, if you have a good season for long cane production, it will do extremely well for long canes. The fruit quality is not outstanding all the time, but it's extremely reliable. It is what my colleagues in Spain call a sort of hard-working variety for them. And there is a lot of information for it, but also a lot of the work that is being done in the wet center. Mark's team is doing quite a lot of optimization on it in terms of irrigation and nutrition. We've got some information on chilling hours required, how we're optimizing the production of long cane. And this year we're doing some trials on dormancy breaking for low chill conditions, which are not as relevant here, but since the variety is now being grown increasingly in Mexico and in Spain, it is important. And I just want to finish with a slide of two of our new advanced selections that growers have been looking at. The one The one on your left is actually been with growers for 2 years, and I've got some reasonable hopes of it because it's always very good fruit quality. The other one is a little bit newer to growers. They only had quite late plants this year to look at, but the fruit size and the ease of plugging of those really long conical fruits, which is quite unusual for fruit of that shape, has been very appreciated. Those fruits there on that hand are all over 11 grams, which as you can tell, I'm quite pleased with that. So I thought I'd leave you with my 11 grams raspberries in your mind. And with that, if you have any questions, I'll be delighted to answer them, provided I've got time.
Thank you so much, Feli. I can just tell you that I'm fortunate to share a building, an office with Feli, and those of us that work there get the opportunity to sample lots and lots of her varieties during the summer, and some of them are just outstanding in terms of flavour. Feli, we are running a little bit over. You have got several questions from guess who? Janet Allen. So Janet always has lots of questions for you. Can I ask you perhaps to respond to Janet's questions in the chat?
Yeah, I'll get to them on the chat. Thank you, Scott.
And thanks to Papp-Rupar, who has also replied to some of the previous questions. So if anybody's looking for answers to those questions, do keep looking in the chat facility. Thank you for now, Feli.
Thank you.
And we now have Alin Borleanu. Alin is the technical officer for Malling Fruits, and Alin is always out and about throughout every season, monitoring and looking at the performance of lots of the Malling Fruits portfolio varieties. So Alan's going to share some of his latest findings from the 2023 season. Good morning, Alan.
Oh, hi, Scott. Not sure if you hear me.
Yeah, we can hear you and we can see your slides perfectly. So the floor is yours.
That's good. Well, thank you very much for your introduction. Good morning, everyone. So as Scott was mentioning, today I'll try to share some information on a couple of our varieties from the Malling Fruits portfolio. It would be quite difficult to mention about each of them in the short time I've been allocated. So I'll be focusing mostly on Malling Ace and Malling Bella. But before going into that, I just briefly wanted to remind ourselves what Malling Fruits stands for. So the brand Malling Fruits has been launched in 2021, and it represents the branding of fruit varieties being commercialized from different breeding programs that we have at NIAB, specifically at East Malling. So Malling Fruits is well known internationally. Varieties from its portfolio are being grown on almost every continent, and this is expanding every year. We also interacting with international breeding programs, and we have different partnerships across the globe. So within the portfolio, we can find varieties of soft fruit, top fruit, and stone fruit, which, as I said, are very well known around the world, and they are serving the needs of the different markets. With NIAB is committed to ongoing agronomic and technical development for our varieties post-release. And for that, we have been and we are working with growers, propagators, research and development centers, but also other stakeholders to develop accurate and robust technical guidelines to help and to help get the best out of each one of our varieties. Perhaps you may have heard recently one of our breeding programs, the strawberry breeding program, including the varieties listed on the screen, have been acquired by Bayer, who has expanded its presence in the fruit and vegetables market to include strawberries. So this move will allow NIAB to concentrate more on its soft fruit research, with a focus on more practical real-world solutions for farmers, consumers, and the environment. It will also open new opportunities to work with leading soft fruit breeders and propagators across the industry, including Bayer. So hopefully, with the Bayer's acquisitions, the main objective will be to enhance the program by leveraging precision breeding capabilities, therefore building upon a NIAB established foundation. So this initiative seeks to advance the quality of strawberries for growers and the entire value chains. So Bayer aims to benefit UK growers while making sure that these great tasting strawberries will reach a wider global audience. However, with all that, Malling Fruit's portfolio still has an abundance of varieties, including some older strawberries, the raspberries that Feli has just mentioned, plums, and also the well-known rootstocks. So going forward, I'm going to touch and I'm going to present you some data I've been putting together from a few growers that have shared with us some production data throughout 2023. So first, just want to share some data with you on Malling Ace. Malling Ace, it's a high-yielding everbear which has been launched back in 2021. And it has exceptional fruit quality, great fruit size, which stays consistent throughout the production season, which is firm, sweet, and juicy. It's very well presented for fast picking. In fact, picking speeds have been on average around 40 to 50 kilos per hour, which is really good for the variety. So Malling Ace is very well suited for glasshouse and tunnel production. You can see there on the left a production of Malling Ace in glasshouse and a production of Malling Ace in the tunnels. This is actually at the wet center this year. And we can see our colleague here, Trevor, being very happy and excited to present some of the food to the House of Lords Select Committee for Horticulture. Unfortunately, I don't have any data, any production data to share from this sort of commercial glasshouse production. But I will share some data coming from a grower in Kent. So they've been planting Malling Ace they've been working with Malling Ace for the last, let's say, 3-4 years. So they started with advanced trials, and this year, I think it was the second year when they were growing Malling Ace commercially. So they've been propagating their own heavy tray plants, 250 cc, and that was in 2022. And they planted them on the 10th of March this year in one of their fields in Merriort. So they decided to go with a plant density of 4 plants per meter. And that's primarily due to the sort of susceptibility to powdery mildew that Malling Ace is having. So we, through the agronomic trials we've done, we've realized that by reducing densities can benefit Malling Ace a lot. So in this case, Hilo Farms have gone at 4 plants per meter. So first pick date in this field has been on the 28th of May, with the last pick date on the 1st of October. And we can see there the production trend line throughout the season. And we can see there's been a first flush sort of end of May, beginning of June. There was a bit of a break at the beginning of July. And then we had the big bulk of the crop throughout August, September. So the quality of the fruit was very good, almost 100% first class. And in total, they were yielding just over 2 kilos per plant. So for that, they were following a conventional spraying program, sometimes spraying twice a week with the first round of conventional and the second round of potassium bicarb. They were feeding at a low EC throughout the whole season, around 1.2, just being careful not to have overgrowth, and which will allow for sort of young tissue, therefore being more susceptible to powdery mildew. So they've been on top with their cultural measures. So climate conditions are very important in the early season during establishment. You need to be proactive with the vents, with the grass, with the weeds management. If you get behind on this, then you give opportunities to powdery mildew to sort of develop, therefore, higher risk to the plants. So they've only done one deleafing throughout the season. So Molling Ace is a very sort of compact variety. It doesn't, it's not very vegetative, so it doesn't need too much managing in terms of removing old leaves. So therefore, it's very, very cheap to grow. We believe that a lower density at around sort of 5 plants per meter is ideal for Molling Ace. Anything above 5, definitely more than 6, will sort of won't be good for Molling Ace due to that sort of powdery mildew susceptibility. Another important aspect from these years, plant quality is very important. So as I said, Hewlett Farm have produced their own plants, so they've been very thorough with the cleaning in the propagation phase. So this demonstrates that a new variety needs a tweaking in order to access its sort of maximum potential. So one of my colleagues in Belgium at one of the research and development centers was saying that the issue with the industry at the moment is that every grower has their own box. When new varieties are coming, they're trying to fit these varieties in that box. Whereas we think the approach should be growers should have more boxes. In fact, they should have a new box for each new variety in order to understand if the variety is good for them or not. Going forward, I'm just gonna share some data on Molling Bella as well. So as Feli was saying, Molling Bella was commercialized in 2017. And it's a mid-season primocane, well suited for double cropping. And it's very excellent in terms of long cane production, and you'll see that in the next few slides. It has an attractive, firm and glossy berry with very good shelf life. So touching a bit on the Spain production, unfortunately, I don't have any production data from this year because when I was visiting Spain a few weeks back, they were just picking, so the data wasn't ready to share. But what I wanted to mention about Spain, so they had a really difficult summer throughout 2023. So lots of varieties have been struggling, even Molling Bella. However, Molling Bella has bounced back quite well. And at the time I was taking these pictures in beginning of November, the crop was looking really, really good. Which shows the resilience of the variety to arid conditions and shows that sort of future proofing against climate changing. So to give you an example of what Molling Bella can do, so this is some data we have from one of our growers in Poland. So in this case, Fieldstone have been planting long canes, I think in this case coming from a propagator in the Netherlands. They've been taken out of the cold store between 15th and 20th of April. They were planting at 1.5 pots per meter, so that was 3 canes per pot, so 4.5 canes per meter. They started the first pick on week 25, and they finished the long cane production at week 39, so that's this sort of blue curve. And then they allowed some growth through, from which they started picking in week 35, and they finished picking in week 45. So in terms of production, for the long canes, they were producing more than 5.5 kilos per pot, so that's close to 2 kilos per cane. And in terms of the primocane, they were doing around over 500 grams per pot. So in just to give you in context, I think over 5.5 kilos per pot there. That means almost 32 tons per hectare of long cane crop. So just to refer back to what Feli was saying, Malling Bella is an excellent sort of long cane production variety. Again, Malling Bella, this time with a grower in the UK, WB Chambers in Kent. So they've been growing long canes, which they've taken out on the 30th of April. They've been planting 2 pots per meter, 2 pots per pots, two canes per pot, so that means 4 canes per meter. They started picking week 26, the long cane, so we can see this sort of blue line, and they finished week 37. And then they started the primo cane week 36, and they finished week 45. In terms of production, in this case, the production was a bit lower. So we can see a total production of 2.4 kilos per pot. That means over 1.2 kilos per cane. And in terms of the primo cane, this was a bit higher, just over 800 grams per pot. So the sort of difference in yield from the UK to Poland could be to a lot of factors, could be the different climate conditions, could be the different plant material each of the growers was using, and so on. But in this context, Malling Bella in the long cane season was producing just over 20 tons per hectare, and in total, adding the primo cane production at the end of the season, I think the production went close to 27 tons per hectare, which shows the potential of Malling Bella as a sort of double cropping variety. So as a take-home messages from today, so as I said, Malling Fruit is the commercial branding for varieties released from various breeding programs from NIAB Malling. NIAB will be focused more than ever to deliver practical real-world solutions for farmers, consumers, and the environment. As I've mentioned, we're embarking on a new adventure with the sort of strawberry breeding program soon being part of Bayer, which will open doors for enhanced innovation in strawberries, promising better varieties for UK growers and a wider global market. Also, as I've shown, Malling Fruit's portfolio has industry-leading varieties, which play a crucial role in supporting growers amid challenging environmental and commercial conditions. Thank you very much. If you have any questions, I'm happy to take them now or later.
Thank you, Alin. Very, very quickly, because we are running a bit behind. I just want to make the point that Malling Ace was planted at the wet centre East Malling this year. And there have been discussions about its susceptibility to powdery mildew. Do you want to just say a word about that at the wet centre? Because I think they were planted at four plants per metre there, and I think we got good control of powdery mildew throughout the season.
Yes, so at the wet centre, we've been planting the plants at 6 plants per metre, and we did have good control of powdery mildew. I think for next year, the wet centre has decided to follow a similar example to Hewlett Farm. So they will be planting 4 plants per metre versus 6 plants per metre, just to showcase a comparison between the two different densities. I think overall, Malling Ace, it's a variety that takes advantage very good of the space you're offering. So it's filling very well the space, and it's able to produce more yield per plant due to that.
Okay, thank you. I just wanted to update that and clarify that, because I think that's been quite a useful exercise for this year. Alin, thank you. We must move on. So thank you to Alin. As I said, we've rejigged the programme slightly because Matt had to leave us earlier, but we're back into disease control now. Our next speaker is Samantha Lynn. Sam has been a CTP PhD student with us at East Malling for the last 3 years, and some of you will have heard presentations from her before. She's been working on genetic resistance to powdery mildew. And Sam, the floor is yours. Please bring us up to date with what you found at the end of your PhD studies.
Okay. Can you see my screen okay?
We can see you and hear you fine.
Oh, here we are. That's it.
We're in presentation mode. Thank you.
Yeah. Okay. Hi. Yeah, I'm here to talk about my PhD project that I've been working on over the last 4 years. And it was mainly focusing on identifying genes that confer resistance to strawberry powdery mildew. And this was all performed at NIAB East Malin. And so I'm just powdery mildew is a worldwide problem in commercial crops. In strawberries and raspberries, it's caused by the pathogen Podosphora fenis, which is from the Rhizopus family, which is an obligate biotrophic fungi that relies solely on its host for survival, whereby it extracts essential nutrients. So the strawberry powdery mildew fungi overwinters in debris and old leaves, structures called Casamathea, which contains ascospores that are released in favorable spring conditions. And this begins fresh infection on young leaves. And once they're on the leaf, they then produce the conidia, and they produce conidia chains, which is here, which rise above the hairs on the strawberry leaf, and then the conidia is then dispersed by the wind, then infect nearby hosts. They undergo rapid asexual reproduction during the summer months and the autumn months, with the optimum temperature ranges for infection between 15 and 25 Celsius, and the humidity levels between over 75%. And once the conidia have actually settled down onto the leaf, then this can then take about 7 to 14 days for it to germinate and to have the cycle repeat. So the switch from the open field production into polytunnels has provided a warm and humid atmosphere to allow strawberry powdery mildew to prevail in modern cultivation conditions. So the powdery mildew infection on the leaves is observed as a white mycelium growth. And this can be on the leaves, the fruit, and the flowers. And this fungal mycelium coverage typically begins on the underside of the leaf, on the young leaves, and then spreads onto the upper side of the leaves. The infection symptoms on the leaf can be seen as an upward curling of the leaf and kind of like a blotching. The dense layer of the mycelium growth on the leaves can cause a decrease in the photosynthesis, and this can lead to reduction in crop yield. And infection on the flowers and the fruit can cause problems with development, and this can cause malformations and even abort the fruit. And this can also lead to severe yield loss due to the unmarketable fruit. In the UK in 2022, 20% yield loss is estimated to be around £75 million. So the commercial control of the disease is through fungicidal sprays. However, growers are limited to the range of active ingredients that can be applied to control the powdery mildew, and there's also concerns of insensitivity and resistance to this method, which is why an alternative solution to the problem is required. So focusing on resistant cultivars offers the most favorable solution to reduce the impact of powdery mildew. The current standard breeding practice to improve disease resistance is to use a slow and extensive disease screening assay. However, by identifying genetic components controlling disease resistance, we can use advanced genetic techniques to identify resistant individuals and upgrade the current system to enable genetic informed breeding. So in the course of my PhD, I was looking at a mildew field experiment, and this is mostly what I'm going to talk about today, but I also will mention a little bit about the strawberry count. So to give you an overview of what a genome-wide association study is, which is a GWAS, so this can be performed to identify a set of genetic variations associated with a trait of interest. We do this by assessing the phenotype and the genotype information. So the phenotype is the symptoms, and here is an example. So you have one that has no symptoms, and you can see the plant with symptoms. And we can analyze the genetic information, and then we can see changes in this genetic information and then identify the location of it. And this can then be provided as a genetic marker to show a more resistant type or whatever it is plant. So for to perform a GWAS, this has incorporated a large amount of varieties and have all of their symptoms assessed, and also to take the DNA and to run that for the genotyping. The data then acquired from both of these can then be applied to statistical analysis to calculate association results to identify genes of interest. So it's with these genes of interest that can be potential markers, but to actually be sure this is what it is, we like to validate it within the laboratory. And so you can do this by using CRISPR-Cas or something to knock out. This would be your gene of interest. You create a vector of your gene of interest that can be put into agrobacterium, that can be transformed into a strawberry. The strawberry is then grown fully, and we can do pathogenesis tests to see if this is actually providing resistance. And then we would have validated gene markers, and this can then be applied to the breeders. So in my project, in the GWAS study, the field work was done all at East Malling, and the goal was to identify resistant genes from powdery mildew. So this involved 331 different strawberry cultivars of commercial importance. Each cultivar had 5 replicates, which totaled 1,655. And these were planted in a randomized design to account for any variation in the soil or nutrients or shade. And the phenotyping was done for the year 2021 and 2022. So looking at the symptoms, I used a 5-point scale. As you can see here in the top left, you have this will be a score of 1, which has no symptoms. And in here it would be a score of 3, which has the obvious leaf curling and also these dark patches that occur. And a score of 5 would actually be leaf necrosis and partial leaf death. So you can see that in this is the scores for 2021, and you can see that it's most of them in July are actually mostly showing no symptoms. And this is probably due to the fact that it was a much milder start to the year. But as you can see, as the season progresses and the temperatures and the humidity rises, this grows. And then by September, you can see that over 50% of them showed powdery mildew symptoms. So then with the data collected could then be collated, and we could look at the different levels of resistance between the varieties. So here I have resistance to susceptible, and you can see that San Andreas is very resistance type. And whereas you go along, you can see that Portola is it's more susceptible to the powdery mildew. So once all of the data was collected, then could do a full statistical analysis, and the results showed that had a high broad sense heritability, which was calculated over 80% for both years. And this shows us that a significant proportion of what was seen in the variation was due to a genetic factor. So that means we're potentially going to see a genetic cause with the powdery mildew. So here I can show you, I have the data that I had, plotted it on a Manhattan plot. And on the outside of the circle is the chromosomes they're placed on. The middle of the circle is for the year 2021, and the inner circle is for the year 2022. And all of the pink dots are significant qualitative trait nucleotides that were identified through analysis. And so the QTN that have been identified are parts of the DNA that correlates with the variation of interest. And as you can see, there were quite a lot of potential QTN that were identified. And but the main thing, and this is the good thing about my project, it was because we had the opportunity to run it for both years, which meant that I found 6 stable QTN, which were actually on in year 2021 and 2022. And it is these ones that we found that would be good to go forward at looking for further validation to be implemented into the breeding programs. And one of these that was actually identified, I think it was 5C, actually had a effect of over 61%. So this is very positive. And then just briefly, just because I had done this GWAS, I also wanted to do an assessment on the flowers and the fruit, and I wanted to find out whether we could identify gene or genes that were associated with the increase in the fruit or flower number that could benefit the growers. And so looked at all the varieties in 2022 and had a look at this, and like this is the same as the Manhattan before, it's just not circular, and the significant marks are in pink, and they are located on the chromosomes at the bottom. And these found several interesting genes that would be interesting to look for in the future to try and validate these genes to maybe to assess it, because this is only taken at one time point. It would be good to try and assess it at several different time points to clarify and validate it in the future. So yeah, so the overall, the development of disease-resistant varieties will support the fruit industry, and this can be done by reducing the animal maintenance cost, lowering the number of fungicides applications, and minimizing the amount of produce that is lost due to the disease. Overall, results from this project provided several potential genetic markers that can be applied to strawberry breeding for disease resistance against powdery mildew. And future focus should be on integrating of these disease-resistant genes into the strawberry breeding program. Thank you all for listening, and I would like to thank the CTP for funding this project, and to my PhD supervisors, Dr. Helen Coxton and Professor Dunwell, and also my industrial representative, Harriet Duncalf, and also the help from Dr. Andrew Armitage. So thank you,
Sam. I'm very grateful to you. I should just say Sam has now moved on. She's finished her PhD. She's now working in the short term with a pharmaceutical company. So thank you so much for taking time out of your new working life to join us.
That's okay.
Because we're running rather late now, we're going to have to move on, but so I'll not offer any more questions for your talk. Thank you again, Sam.
Thank you.
So finally, we have Tom Passey, who's going to talk to us a little bit about Mucor and Rhizopus, which has been causing problems for a number of strawberry growers in recent years. And Tom is one of the pathology team at NIAB, at the East Malling site. Tom, share some news. I'm sorry to put you on the spot, but if you can be reasonably quick, because we are running over time.
No worries. Hopefully you can see my screen and
everything's working perfectly. Thank you.
Okay, cool. So thanks, Scott. So I'm going to be talking today about Mucor and Rhizopus, so I shall get started. So a couple of research projects that we've been working on over the last decade with these diseases. Some work done by a PhD student, Solomon Aguirre, who worked for us for some time, funded by Berry Gardens. Some follow-on work then as an Innovate project, again supported by Berry Gardens, that was work carried out by Ethna Brown, and some current work we're doing with BBSRC. So just as a quick reminder of what Mucor and Rhizopus are. So our main post-harvest rots on strawberry are the Mucor, Rhizopus and Botrytis. Many of you will think of botrytis and be aware of botrytis as the big issue with post-harvest, but actually Mucor and Rhizopus are quite important post-harvest diseases. Sometimes previously lumped in with botrytis as just general post-harvest diseases, but the major difference with Mucor and Rhizopus is they both cause a real leaking of the fruit. So whereas the grey mold of botrytis, the fruit can stay fairly firm, just has this horrible grey mycelium growing over it. The Mucor and Rhizopus species will cause the fruit to really degrade and leak and then dribble over everything from there on. There are differences between the two, although they're both leak diseases and lumped in together quite often. There is a difference between them. Mucor is generally the one that's seen more on strawberry through the season, whereas Rhizopus just tends to be an issue towards late summer and then into the autumn. There's not a huge amount of difference between varieties. Most are susceptible, and there's not a huge difference there. So a lot of the work that Solomon was doing was looking at the epidemiology of the disease. I'm going to go on to some of the work we want to do on prediction, and you can't do that without knowing about the diseases themselves. And a lot of what Solomon was looking at was on germination of Mucor and Rhizopus and infection events. So what he did find was generally at low temperatures and so sort of 10 degrees, at very high temperatures, so 30 degrees, the Mucor and Rhizopus don't particularly like it. They do prefer it in around those mild temperatures around sort of 20 degrees, so 21 degrees here. He was seeing really good germination figures. And also, as with many of our fungal pathogens, the more humid it is, the more they like it. So for Mucor on the top here, it prefers it a little bit more humid than Rhizopus requires it. Rhizopus grows a little bit lower in humidity, but both of them like mild temperatures, high humidity. And then there's a difference between how susceptible fruit is at different ripening stages. So Solomon looked at differences between ripe fruit and what he terms yellow fruit. So these are sort of just underripe fruit. And there is a large difference there. The ripe fruit are much more susceptible than anything below that. And that again is related to humidity, less related to humidity than just pure germination of the spores is. So the fruit can be susceptible at various levels of humidity when they are infected. Bit further into this, so it is the only fruit that can be infected are white and ripe fruit. So anything, flowers, unlike botrytis, where flowers can be infected by botrytis, with Mucor and Rhizopus, even the green fruit stage. So these are the initial fruitlets starting on the plant. They're not susceptible. It's only when the fruit starts getting to a point where you're just getting slight ripening, slight reddening of the fruit. That's when they really become susceptible to the Mucor and Rhizopus. And the more ripe they become, the more susceptible they become. And as mentioned previously, there's no huge difference between different cultivars on this. Most show similar levels of susceptibility. A bit more here on sort of the how high the inoculum level needs to be. And although, as expected, you see here for Mucor, as the number of spores increases if a fruit is inoculated, as you get more rots, but actually you don't need that many spores to cause an infection. So 100 spores here per fruit, and you were getting at least 50% infection by Mucor, and with Rhizopus, that's kind of pushing up towards 75% infection. So you don't need a lot of spores to get infection. a little bit more then moving on from that is infection of attached ripe fruit work that Solomon did here. So what we're seeing here is that actually the figures at the bottom are vapor pressure deficit, and these are kind of all in the normal expected range during the growing season. So this is taking out those low temperatures of 10 degrees and the plus 30 degree temperatures that, although we may be seeing more frequently in season, aren't our normal temperature range. And actually throughout these normal expected temperature humidity scales that we have during the growing season, there isn't a huge difference between Mucor and Rhizopus rots required for infection. And this gives us an issue which I'll go into further in a minute. So we know a little bit more about the diseases, and then the next stage is to start working out, can we actually start predicting when these infection events are going to happen or not? So by this, we set out an experiment that was done in the previous trial, and we're doing again this year with the current BBSRC project, where we are exposing, so we bag fruit before they become susceptible, leave for a couple of weeks, remove the bag for 24 hours and allow infection in that 24-hour period. We also use a selective media to trap inoculum in the same period, and we can then compare the fruit infection to the amount of inoculum that we're seeing on these plates through the season. As I mentioned before, this was done previously in the previous projects in 2018, 2019, and again this year. And what we've seen is, as we kind of expected from previous results, that generally the riper the fruit is, the more infection you see. As you can see here, the red columns are the ripe fruit, and they tend to go, you have higher incidence of infection for those ripe fruits compared to the white fruit. But the other thing we've seen is predicting the exact incidence for both Mucor and Rhizopus is quite hard. Regression modeling here that Xiaoming's been working on, you can see there's perhaps a slight hint of regression in the Rhizopus, but the Mucor is showing no real pattern at all, especially for the ripe fruit. With the white fruit, it's a little bit easier, but probably because there's less observed incidence on the white fruit. But for the ripe fruit, which is the area we're probably most interested in, it doesn't fit easily into regression models. So some further work that has been done then is around this sort of modeling approach, this prediction approach, looking to see if we can predict what is going to happen or not. And the quantity prediction is okay, but it's not great. And so we investigated whether a classification-based approach would be better. By that, this is based on percentage, on significance values, and converted into classes on the incidence of fruit rotting, and putting those into a low, moderate, and high risk prediction. And what we found is that generally the predicted low, moderate, and high generally fit with the observed level. So you can see here 125 low predictions were predicted low and were low. And generally the moderate, and the high are the highest numbers in those columns. The issue is when you have a low prediction and a high observed level. So it only happens on 9 occasions here, but those 9 occasions could be very problematic if you're only going with a prediction system on when to try and keep on top of these diseases. So this is for Mucor, and just to I'll very briefly show this, but this is the binary decision tree that is used to make these predictions. And if anyone has any burning questions on this, please get in touch with myself or Scott, and we can put you in touch with Xiaoming, who's developed this, and would be more than happy to talk to people on it. The Rhizopus shows similar figures to the Mucor. So again, Generally, most of the predictions are low infection. Most of the time there is a low infection, and that is equal to the observed. On some occasions it is predicted high, and it is high. But again, the issue is where you have a low prediction, but actually a high level of a high observed level of infection. Again, the binary decision tree this time for the Rhizopus. Okay, so going through the conclusions of this work then, the dependency of infection for Mucor and Rhizopus on climatic conditions isn't stringent. So the problem that this thing gives us is that it makes it very difficult to really make predictions on the most likely infection times. Really accurate inoculum quantification in multiple locations across a field would be essential for accurate prediction. This probably isn't feasible to really be able to get a good idea of where the high spore infection efficiency is across fields. It's going to be very difficult to do in those multiple locations. And given the rapid lesion development, lack of products controlling these two pathogens, whether the present models with model accuracy are really of any significant value for production comes into question. The main thing then to really look for with these two pathogens is hygiene and cool chain. As with most of our post-harvest diseases, cool chain is absolutely imperative. The sooner you can get fruit from the field into the cool chain process, and the more robust that cool chain process is, the less likely you're going to have post-harvest issues. But again, hygiene is highly important. If you can make sure that you're not leaving overripe fruit on the plants, not having dropped fruit on the ground, and getting all that waste fruit that's picked off as far away from your plants as you can, that is the most important thing. And hopefully with SWD control, this is obviously something that you'll hear a lot from Michelle Fountain and my other entomology colleagues, that hygiene is highly important. And so with SWD, the same can go for these post-harvest diseases as well. Okay, Scott, I tried to go as fast as I could for that. Just some thank yous again, Dr. Solomon Aguirre, Ethna Brown, who is now Dr. Ethna Brown, and a lot of the work done on this in all the projects has been done by Joyce Robinson. Thanks, Scott.
Thank you, Tom. You've done well. Thank you. Yeah, slightly disappointing at this stage. Is there any scope to do further work to try and improve this, or do you think that it's going to be too difficult?
It's going to be hard because it's so susceptible at different across so many different sort of climatic situations. It's hard to really pinpoint very accurately down to when the biggest risk is. As those tables showed, you can predict the risk quite accurately for most of the time, but there's always that problem with there's only any point putting in predictions, putting in prediction models for your spray regimes if you're not going to have those I've predicted low and then you get high, because that's when all of a sudden you send that crop to market and it all comes back because it's potentially got issues. So there's potentially work in other areas we can do, and they are important diseases, but cool chain and hygiene at the moment are the most important takeaways for growers.
Okay, understood. Thank you. Thank you so much, Tom, and thank you to all our speakers from session 2.